Welcome back, everyone. Ready for another deep dive.
I'm ready. Let's do it.
Today, we're going to explore a case from the world of veterinary neurology, something pretty unique.
Definitely unusual.
We received a research article from one of our listeners about a dog diagnosed with a really weird disseminated infection.
Interesting.
It was caused by a fungus called Schizophyllum commune, and the crazy thing is it even reached the dog's brain.
Oh, wow. That's not something you see every day. So we're talking encephalitis caused by S. commune.
That's exactly what we're talking about.
The first reported case in a canine, actually. It's a pretty groundbreaking case, published in the Journal of Comparative Pathology.
Okay, so we're going to break this all down, but before we get too deep, let's just, start with the basics. What can you tell us about this particular dog?
Well, this case involved a 10-year-old mixed breed dog.
Okay, a bit of a senior dog then.
Right. And he presented with a whole bunch of symptoms, but the most concerning ones were neurological.
Neurological issues. So, like, what kind of things are we talking about?
Things like a waddling gait, trouble walking.
Okay, yeah.
And decreased postural responses, which means he had difficulty maintaining his balance.
So basically something was off with his coordination and balance?
Exactly. The vets initially thought it might be a cranial mediastinal neoplasm.
Okay, hold on. Break that down for me a little. Cranial mediastinal what?
Neoplasm. Basically a tumor in the chest area.
Ah, okay. So they thought it might be a tumor.
Yeah, especially because they found a large mass in the chest and the dog's lymph nodes were enlarged. They were worried it might even be spreading to other parts of the body.
Systemic metastases.
You got it. But then things took an interesting turn when they did a biopsy.
What happened with the biopsy?
Well, they did fine needle biopsies of the mass and found something unexpected, fungal hyphae.
Fungal what now?
Hyphae. They're basically the long, branching, filamentous structures that make up the body of a fungus.
Okay, so they found evidence of a fungus in the mass.
Right. So now, instead of just a tumor, they had to consider a possible fungal infection as well.
Okay, so at this point, they've got this dog with neurological problems, a large mass, and now they're finding evidence of a fungal infection. What do they do next?
They decided to do some imaging to get a clearer picture. They did a CT scan and an MRI.
Classic imaging tool. So what did those scans reveal?
Quite a bit. The CT scan showed osteolysis in the second rib and one of the vertebrae.
Osteolysis meaning?
Bone destruction. Basically, the fungus was eating away at the bone.
Yikes, that sounds serious.
It is. The CT scan also showed the cranial mediastinal mass we talked about, and it was pretty big, like 7 centimeters in diameter.
Wow, that's a substantial mass. And the MRI, did that show anything?
The MRI showed something even more concerning, actually. It revealed lesions in the white matter of the brain.
Lesions in the brain? That's definitely not good.
Not good at all. Specifically, there were lesions in the right frontal lobe and the left cerebral hemisphere. And these lesions lit up on the contrast-enhanced T1-weighted images, which means they were areas of active inflammation.
So not just lesions, but active inflammation in the brain.
Yeah, definitely a red flag. The imaging basically blew the initial suspicion of a simple tumor out of the water.
Right. It suddenly became a much more complicated and worrying picture.
Exactly. Now, the question was, what was causing those lesions? Was it the fungus?
That's the million-dollar question, isn't it?
Absolutely. And to answer it, they needed to figure out what happened next. Sadly, the dog's condition just kept getting worse.
Oh, no.
And unfortunately, he passed away. But even though it was a sad outcome, the vets were able to perform a necropsy.
A necropsy?
Basically an autopsy for animals. It's how we investigate the cause of death and really understand what was going on inside the body.
Okay, so they did a necropsy. Did they find anything significant, especially in the brain?
Oh, they definitely did. They did. The necropsy revealed some pretty critical visual evidence, especially when they looked at the brain.
I bet. What did they see?
Well, from the outside, the brain had this milky white surface, which is already a little strained.
Milky white. That doesn't sound healthy.
Not really. And then when they looked at cross-sections of the brain, the corticomedullary junctions were indistinct.
Remind me what those are again.
Sure. It's the area where the brain's outer layer, the cortex, meets the inner white matter. It's usually a pretty clear boundary, but in this case, it was all blurry.
Oh, I see. So something was definitely disrupting that normal structure.
Exactly. And the white matter itself looked discolored. But the real clincher came when they examined the tissue under a microscope.
Okay, what did they find under the microscope?
They found granulomatous inflammation in multiple organs, including the brain.
Granulomatous. Okay, can you break that down for me? What exactly are granulomas,
Yeah.
and why are they significant?
Right. So granulomatous inflammation is a specific kind of inflammatory response. It's how the body tries to wall off an irritant.
Like a little fortress built around something it doesn't like.
Yeah, kind of. In this case, the irritant was the fungal infection.
So the dog's body was trying to contain the fungus.
Exactly. And when you look at these granulomas under a microscope, you see these clusters of specific immune cells like epithelioid histiocytes and multinucleated giant cells. They're all working together to try and contain the infection.
Wow, that's amazing. But if these granulomas were in the brain, that means...
That the infection had spread beyond just the initial mass.
Right.
In fact, they found granulomas in the cranial mediastinal mass, the bone lesions, lymph nodes, heart, kidneys,
Whoa, all over the place.
lungs, and even the esophagus.
So not just a localized infection, but a full-blown disseminated infection throughout the body.
Exactly.
Okay, so we've got these granulomas, and we know the infection spread. But how did they know for sure that it was Schizophyllum commune?
That's where DNA analysis comes in. They took samples of the formalin-fixed tissue.
Formalin fix.
Yeah, it's a solution used to preserve tissues. Anyway, they extracted the DNA from those samples and did PCR and sequencing to pinpoint the fungal species.
Okay, back up for a second. PCR and sequencing.
Right. PCR stands for polymerase chain reaction. Think of it like a molecular photocopier. It amplifies specific regions of the DNA, even if you only have a tiny amount to start with.
Okay, makes sense. And sequencing.
Sequencing is basically figuring out the order of the building blocks in the DNA, the nucleotides. It gives you a unique fingerprint for each species.
So they were able to identify the fungus based on its DNA fingerprint.
Exactly. And the results showed, without a doubt, that it was Schizophyllum commune. Knowing the specific species is super important because different fungi can cause different types of infections. It helps guide treatment decisions.
That makes total sense. Okay, so now that we know who the enemy is, let's talk a bit about Schizophyllum commune itself. Most people probably haven't heard of it. What is this fungus, and where do you typically find it?
It's not exactly a household name, that's for sure. Schizophyllum commune is a type of fungus that's found all over the world. It usually grows on decaying wood, like fallen logs or tree stumps.
So pretty common in nature then.
Oh, yeah. It loves environments that are rich in organic matter and moist.
Makes sense. So it's out there in the environment, but can it infect humans too?
Yes, it can. It can cause infections in people with healthy immune systems.
So not just those who are immunocompromised.
Right. Anyone can get it. It's usually associated with respiratory problems in humans, but in people with weakened immune systems, it can sometimes cause more serious infections.
So this fungus can cause problems in both humans and animals. That's not good.
No, it's not.
What about brain infections, though? How common is it for S. commune to cause infections in the brain, especially in humans?
Brain abscesses caused by S. commune are actually pretty uncommon in humans.
So this dog's case is even more unusual.
Yeah, it really is. It highlights the serious nature of this fungal infection. To understand the full significance of this case, though, it's helpful to look at what we know about other animal infections.
Okay, let's do that. Where do we even begin to look for that kind of information?
Well, the research paper actually includes a table, Table 2, that summarizes other reported cases of S. commune infection in animals.
Interesting. What does that table tell us? Any patterns or common themes?
There are definitely some patterns. It seems like S. commune has been found in a few other animal species. For example, there were a couple of cases in cheetahs and one in a harbor seal.
Cheetahs and a seal. Wow, that's a diverse group.
It is, and what's interesting is that a lot of those cases involve the respiratory tract, which makes sense since that's the primary way the fungus gets into the body.
Right, through inhalation of spores, most likely.
Exactly. But what makes this dog's case stand out is that the infection spread to the brain and caused those severe neurological symptoms.
So respiratory involvement seems to be a common feature of S. commune infections. But this dog's case, with the brain involvement, is pretty unique. It begs the question, how did the fungus spread so effectively throughout this dog's body?
That's a great question, and that leads us to the next big topic, the dissemination pathway. Based on everything they found, the researchers think the dog probably inhaled S. commune spores from the environment.
So it started in the respiratory system.
Most likely. From there, it probably colonized the lymph nodes in the chest area. And then eventually it got into the bloodstream.
And once it's in the bloodstream.
It can basically go anywhere.
And that's what happened here.
Yeah. They think the fungus traveled through small blood vessels in the white matter of the brain, leading to those lesions we saw in the MRI.
And those lesions, of course, messed with the normal neurological function, leading to the waddling gait, the decreased postural responses, all those neurological problems the dog was having.
Exactly. It's a really good example of how an infection that seems localized can actually become a systemic problem.
Well, with some pretty serious consequences.
Absolutely. It highlights why it's so important to understand not just the pathogen itself, but also how it spreads within the body.
Speaking of understanding the pathogen, we talked earlier about how tricky it can be to diagnose fungal infections. Can you talk a bit more about the challenges involved and how this case really emphasizes the need for advanced diagnostic tools?
Definitely. Diagnosing fungal infections can be tough, especially in cases like this where the symptoms are so unusual. Traditional culture methods where you try to grow the fungus in a lab can be slow.
Slow, meaning?
It can take days or even weeks to get results, and they're not always reliable. Sometimes the fungus just won't grow, even if it's there. And when you're dealing with a critically ill patient, you don't always have that kind of time.
So you need faster and more accurate ways to diagnose these infections.
Exactly. And that's where molecular techniques like PCR and DNA sequencing come in.
Those are game changers, aren't they?
Oh, absolutely. They've completely revolutionized how we diagnose all sorts of infectious diseases, but especially fungal infections.
We talked about PCR and sequencing a bit earlier, but can you remind me how they work again?
Sure. So PCR, which stands for polymerase chain reaction, basically amplifies specific regions of the DNA. It's like taking a tiny sample of DNA and making millions of copies of it. And sequencing is figuring out the order of the building blocks, the nucleotides, in that DNA.
Okay, and that sequence is unique to each species, right?
Exactly. It's like a fingerprint. So in this case, they were able to definitively say that the fungus was S. commune based on its DNA.
So the DNA evidence was the clincher.
Exactly. It allowed them to gain a deeper understanding of how this particular fungus behaves in animals, which could help with future cases like this. Without these advanced techniques, they might never have figured out what was going on.
It's amazing how technology is changing the face of veterinary medicine, allowing us to dive deeper into these mysteries and make more informed decisions.
It really is incredible. This case is a perfect example of how these advancements are changing everything.
All right, so we've talked about the patient, the fungus itself, the challenges of diagnosis, and the power of those molecular techniques. Now let's shift gears and focus on the bigger picture. What does this case, the first reported case of S. commune causing encephalitis in a dog, mean for vets out there?
I think it's a wake-up call for the veterinary community. It makes us realize that S. commune, which we used to think of as mainly a respiratory problem, can cause serious neurological issues.
So it's challenging our assumptions.
Yeah, we have to broaden our thinking when it comes to these kinds of infections.
So what are the main takeaways for vets in terms of diagnosing and treating these infections?
I think the most important thing is to keep an open mind. Vets need to consider S. commune as a possibility, even when they see neurological symptoms that don't immediately seem related to a fungal infection.
So be more proactive and consider a wider range of potential diagnoses.
Exactly. And early diagnosis is always key, but especially for fungal infections, which can be really hard to treat.
So early intervention is crucial. Are there already established treatments for S. commune infections, maybe based on what we know from human cases, that vets can use for animals?
There is some knowledge about treating S. commune in humans, but it's not so simple to just apply that to animals. Animals and humans can metabolize drugs differently, the side effects might be different, and the location and severity of the infection can also impact treatment decisions. So while human cases can give us some ideas, we really need more research to figure out the best ways to treat S. commune infections in animals, especially those that have spread to multiple organs like in this case.
So we need more research to bridge that gap between human and veterinary medicine and develop more targeted treatments for animals.
Exactly. That's the burning question right now.
And how does this particular case contribute to that effort?
Well, this case study is a great starting point. It raises awareness, it gets people thinking about new research questions, and hopefully it will inspire more research into better treatments for these types of fungal infections. It's a step in the right direction.
It's a reminder that we're always learning and adapting as new challenges arise.
Definitely.
Okay, so the research paper also goes into some pretty detailed descriptions of the fungus itself, particularly its appearance under the microscope. Can you walk us through some of those observations?
Oh, sure. Remember those granulomas we talked about, the clusters of immune cells? Well, when they looked at those granulomas under the microscope, they could actually see the fungal hyphae inside them.
Hyphae.
Yeah, those are the thread-like structures that make up the body of the fungus. The researchers described them as hyaline, which means they were transparent or colorless, with septate hyphae.
Septate hyphae.
Yeah, it means the hyphae have these little cross walls inside them, kind of like partitions, giving them a segmented appearance.
It's amazing how much you can learn just by looking at these tiny structures under a microscope.
I know, it's fascinating. And these septate hyphae had a distinctive Y-shaped branching pattern. That was another clue that pointed towards S commune. But to be absolutely sure, they needed to go a step further and analyze the fungal DNA.
And that's where those molecular techniques came in, right? PCR and sequencing?
Yep. They extracted DNA from the tissue samples and used PCR to amplify certain parts of the fungal DNA. And once they had enough DNA, they sequenced it, which means they figured out the order of all the nucleotides. And that sequence matched up perfectly with S commune.
So the DNA evidence sealed the deal, providing that final confirmation.
Exactly.
It really highlights how powerful these techniques are.
For sure. It's like having a super-powered magnifying glass that lets us see the hidden world of these microorganisms.
Okay, so the paper also mentions some of the challenges they faced when working with the tissue samples, specifically that they were formalin fixed. Can you explain why that's a challenge?
Yeah, so formalin is a solution that's used to preserve tissues, but it can be a bit of a double-edged sword.
How so?
Well, it's great for preserving the structure of the tissue, but it can damage the DNA over time. So when you're trying to extract DNA from formalin-fixed samples, it can be tricky to get good quality DNA.
Ah, I see. So it's essential for preserving the tissue, but it can degrade the very thing you need for DNA analysis.
Exactly.
So how did they get around that problem?
They used special DNA extraction kits and PCR primers designed to work with DNA that's been a bit damaged. Think of it like having specialized tools for a really delicate job.
Clever. So they were able to extract the DNA and get the information they needed despite the challenges.
Yep. It's a good example of how resourceful researchers can be.
Okay, before we move on, I want to touch on one more thing. The collaborative nature of this research. The study involved a whole team of experts from different fields.
Oh, yeah. It was a really great example of interdisciplinary collaboration. You had veterinary pathologists, surgeons, mycologists, molecular biologists, all working together to figure this out.
It's like piecing together a puzzle, isn't it? Each person brings a different piece.
Exactly. This case really highlights how powerful it can be when experts from different areas team up.
All right, so we've covered a ton of ground today. We've talked about the microscopic details of the fungus, the challenges of diagnosis, the importance of molecular techniques, the implications for veterinary medicine, and the power of collaboration. We're going to take a short break, but when we come back, we'll wrap up our deep dive into this fascinating case of Schizophyllum commune.
And we're back, ready to finish up our exploration of this unusual Schizophyllum commune case.
It's definitely been a wild ride learning about this fungus and its surprising trip into a dog's brain.
You know, before the break, we were discussing the teamwork aspect of this research. It took a real village, so to speak, to pull all the pieces together. Absolutely. This case was a prime example of how experts from different fields you had, your pathologists, surgeons, mycologists, molecular biologists, can collaborate and solve a real medical mystery.
And that collaboration really paid off, leading to a much deeper understanding of this case than any one specialty could have achieved alone.
It really did. The picture that emerged, though, is both fascinating and kind of scary, right?
Fascinating because it expands our knowledge of what S. commune can do.
Right, but scary because it highlights the potential for these kinds of fungal infections to cause serious neurological problems in our furry friends.
Absolutely. So as we wrap up, let's bring it back to our listeners for a moment. What are the key takeaways here? What should people, especially pet owners, be thinking about in light of this case?
I think the most important takeaway is that fungal infections can be really serious, even life-threatening, and they're often overlooked.
Right, they can fly under the radar, so to speak, because the symptoms might not scream fungus right away.
Exactly. So if you notice any unusual symptoms in your pet, especially neurological ones, don't hesitate to ask your vet about the possibility of a fungal infection.
And don't be afraid to ask about those advanced tests we discussed earlier, like PCR and DNA sequencing.
Those can be super helpful in pinpointing the cause, leading to a quicker diagnosis.
And this case also reminds us to pay attention to our pet's surroundings, like decaying wood, where S. commune can be found.
It's a good reminder that our pets encounter all sorts of things in their environment, And sometimes those encounters can have serious consequences.
So being mindful of their surroundings and potential sources of infection can help protect their health.
Definitely.
Covered a lot of ground today, from those tiny fungal structures all the way to the bigger picture of veterinary medicine. It's been a journey of discovery.
It has. And while this particular case didn't have a happy ending, the knowledge we gained from it will hopefully help other animals in the future.
It reminds us that research is ongoing and we need to stay vigilant, especially as new threats emerge.
Yeah.
But it also highlights the amazing progress we're making in veterinary medicine, thanks to the dedication of researchers and vets and the power of these new technologies.
Couldn't agree more.
It gives us hope for earlier diagnoses, more effective treatments, and ultimately, healthier and happier lives for our furry companions.
From Vet NeuroJar, keep those minds inspired, hearts light, and tails wagging.